Welcome, Professor.
Pleased to be here.
Tell us a little bit about your laboratory and your current work.
Well, my current work, and really I should restate, my current interest is not so much my own work as trying to be apprised of the work of several others that I consider to be very important. And that work has to do really with the nature of life. And you say, whatever do you mean by the nature of life? And the answer is that there have been a series of advances in our understanding of cell metabolism. And those advances lead quickly to an enhanced understanding of, in effect, how we live and how we age. And so aging itself, as you are already aware, is a hot topic. Lots of people interested in the nature of aging. But for the most part, it's been very difficult to grapple with the concept of aging itself and, in effect, changes over time. And now the difficulty continues, but there have been a few instances of enhanced or new understanding, and those enhanced understandings, the new understandings, are leading to what I consider to be a revolution in the nature of life, or likely to be a revolution in the nature of life. And you might ask, whatever do you mean? That's sort of vague. And the answer is, just consider for a moment the nature of a cell itself. We have all kinds of cells in our body. You can divide them, if you will, into two great classes. Those that keep reproducing over and over and over again. Skin cells, liver cells, lung cells, pancreas cells, on and on. And then you say, okay, I'm interested in pathology and or health. What's the problems with lung cells, liver cells, and so on? Well, there are a myriad. You can go to a medical textbook and go under any of these topics, liver, lung, and whatever, and have a catalog of potential disorders. But if you say, okay, what is of key interest to most people, besides these highly specialized disorders, my answer would be aging itself. And then you would say, okay, I know I have pictures of myself from high school that don't quite look the same, that my hair has changed color to a degree, on and on. So what is aging? And how is that relevant? And I think there's a series of answers, but the one that's the most telling and I think shows the most promise is an understanding of the various organelles that exist within cells. And then you say, organelle? What's an organelle? And so in the long-lasting cells of the body, those are the ones that I think are of most current interest, and the brain cells, heart cells, cells that you get one time and one time only. Beating in your chest today are cardiac myocytes. Those cardiac myocytes are contracting 50, 60, 70 times a minute. They've been very loyal to you. I don't know your exact age, but whatever it is, they've been contracting at that rate for a long time. And then you say to yourself, okay, that's all nice, but what are you really talking about? Well, the answer would, I think, go to the following. If you look up the constituents of cells, besides specific organelles, you have a host of proteins. And with modern biochemistry, these can be characterized. Well, a typical cell can have 5, 6, 7,000 proteins, a myriad of proteins. And then you say, okay, that's nice. What do? Well, they have specific functions. So again, go back to the heart. You've got myosin derived molecules that shorten, lengthen, and are responsible for the contraction of the heart muscle. And then you say, gee, again, that's nice. I'm happy to hear it. And, keep going, keep doing your thing. But you talked a little bit, or said something about maintenance. And what do you mean by maintenance? And the answer is that there have been a series of remarkable advances in the last few years in our comprehension or understanding of the general idea of maintenance. Now, it's become popular in some ways. So, for instance, I don't think, or at least I'm not aware of a single person of my acquaintance who doesn't have a sense that exercise, at least to a moderate degree, is good. And that every day you ought to move this, move that, and so on, and that it's beneficial to your health. Okay? That's reasonable. But what else? Well, if it's not exercise, it's diet. Diet has become almost a religion. People eat and they put food into their mouth, and as they do it, the little clicking mental qualities say, cholesterol, lipid, oh good, this is an orange, vitamin C. So in a way, we're all doctors, and doctors for nutritional health. And if that's so, then what can these different ingredients do? Well, some are just essential to specific functions. But in addition, the key point that I'm interested in at the moment, and I think is going to be of widespread interest, goes to the concept of cell maintenance. That here, in my case, I'm 84 years old, and if I have some brain cells, whatever they are, they've been around a good long time. Well, if a cell contains these 6,000 or 7,000 proteins, all of us are aware that proteins age, they change, they deteriorate. So the key point of it all is that there's been remarkable advances in our understanding of how a cell survives for decades, and more specifically, the essential requirements for that survival. The key point here is that it's now known that within a single cell, a single brain cell, for instance, there are protein molecules now named chaperones, C-H-A-P, like the person who guards the personal qualities of young people, and that these chaperones have remarkable functions, specifically that they meander through the cell, back and forth, here and there, attach briefly to specific proteins, check the protein for quality control. And if the quality is good, the chaperone releases the protein, and the protein goes swimming on its merry way. If, for some reason, the quality has deteriorated and there's a specific defect that can be discovered, the chaperone can label this protein. And the label, fancy big name, ubiquination, U-B-I-Q-U-I-N, so on, or for those in the trade, UBI for short, a label is put on a protein. That protein label marks that specific protein for destruction. And if the cell is working properly, and this happens now in brain cells, retinal cells, heart cells, that specific molecule is destroyed and replaced. A new copy takes its place. So you have now, in brain cells, heart cells, many other long-lasting cells, a cycle going on. And although it's relatively new, and I should tell you the Nobel Prize in Medicine in the year 2013 was awarded to a man with an unlikely name, Aaron Chechanova, and I have the spelling, I think right, Ciechanover, or at least I'm very close. And then you say, what did Aaron Chechanova do? Well, he showed that one of the qualities of these cells is that they have a specific organelle, the proteasome, and it is the proteasome that takes in an aged protein and destroys it. Actually takes it back to the amino acids from which it was originally synthesized. And this recycling system that is largely representative of long-lasting cells, brain cells, heart cells, retinal cells, is a kind of wonder. And as I said, you can easily see its discovery leading to a Nobel Prize. And you don't need a lot of imagination to think in another level. Imagine that in your life you have taken a different road, a different route, and that you went into a pharmaceutical company, as a trainee, scientist, whatever, and with hard work and high capability rose up. So now here you are in the chairman's office at Merck or Pfizer. You're the boss, you're the big one. Okay, and in comes one of your basic scientists and starts telling you this long tale of renewal, recycling, destruction, renewal, on and on. You'd take a finger if you were someone who is on the strong side of personality, and you'd point it at this person, and you'd say, Marie, or Jeffrey, or whatever the name is, what are we at Merck doing on this? Or what are we at Pfizer doing on this? And the person might say, if this was an earlier time, nothing at all. I'm here to bring you up to date, to inform you of what's going on in cell biology and pathology. Well, as an executive, quickly you'd get the sense of the thing. You'd say, what are you saying? That we have no effort going on in this field at the moment? You're just here to inform me? Well, I'm telling you, get on it. Get on it quickly. Get on it with intensity. I want a group set up. This is the key to aging. This is the key to all sorts of pathologies. That's what you're describing to me. Do it. Do it now. You'll be back here 1 week. I want in writing what efforts you've put forward, what's happening. Thank you for your time. Goodbye. Bang, gone. Okay, so I'm assuming, without any specific knowledge, I get no calls from any large pharmaceutical company say, Alan, let us inform you of what we're doing. I don't know that at all. On the other hand, I think I have common sense and actually high regard for many of these companies. They really have made substantial advances over the decades. Well, if the set of assumptions, surmises I just put forward are correct, and I'm convinced they are, that means that there are active efforts going on that may have started in the year 2013 with this Nobel Prize, may have started before then, but there are active efforts. And just think to yourself that you're a financial whiz, one of the top financial people at one of these companies, and you have an idea that a product might be possible, a pill that would slow aging or prevent some of the deleterious effects of aging. You don't have to call in your salespeople and ask them, oh, give me some estimate of the sales possibility of an anti-aging pill. You know, it's stratospheric. It would go through the roof. If one or another company is the first in and gets a good product that actually delays aging, however little, let's say it's 5, 10%, whatever. I don't have to be a financial whiz or expert to know that the sales would be incredible. You know, who wouldn't take it? Why not? Unless you couldn't afford it. Okay, so if there is then a possibility of an enhanced understanding of the true cell biology of aging, and there are some possibilities for improving the work of the cell such that aging can be slowed, or at least some of the pathological accompaniments of aging can be slowed, then I think we're into a new era. And when I say new era, you should be aware that although this science is new, and again, at least on my part, just partially understood, it's not new in another way. And the best example that it's not new in another way is, well, I don't know if you've ever been to London, England, as a tourist. And one common thing that people often do if they go to London is go outside the city and visit some of the churches. There's wonderful churches, beautifully constructed, 20, 30, 40 miles outside London. Well, if you go to one of those churches, behind the church there'll often be a graveyard. And you may think that you're a settled and mature person, but you go into one of those graveyards and look at some of those headstones, and I defy you not to get a tear in your eye, because those headstones will date back to 16, 20, 17, 15, whatever. And you will see a headstone. Little Margie Smith died age 5 of the fever. Little Billy Wolf died age 7 of the fever. On and on. Children dying at these horrific young ages. You know, there were no antibiotics. And remember, in England of that era, no refrigerators, no way that was really useful of preserving fresh fruits and vegetables. So vitamin C, vitamin others, and short supply during the winter. Well, and again, if you're a rapidly growing child and you don't get an adequate supply of nutrition, especially vitamins, then you're all the more susceptible. Second, England then was a far poorer place, and poverty has some equivalents. One of the equivalents of poverty, and you'll see it now today, and still in the United States, is crowding. You know, you may be particularly fortunate, and when you go at night to go to sleep, you likely have in your apartment or in your house an adequate private space. Well, that wasn't true 600 years ago in England. People, cheek by jowl, crowded together. And so again, if one child gets a cold, cough, cough, it goes on the other and on and on. So things of that type would spread like wildfire. Anyway, so there's been a dramatic change in public health on a variety of levels, and not to mention gender and immunizations and all. So the world has gotten to be a far safer place for children, thankfully, and even to a degree for adults. And the lifespan keeps increasing slowly up, but there certainly is a dramatic change from England in the year 1100 or 1200.
Tell us about the implications of this for macular degeneration.
There is another specific problem that I don't think is quite as clear. But you can make an argument that macular degeneration, and the one that is best known, most common, most feared, is so-called age-related macular degeneration, ARMD in the code. And ARMD has the following qualities. If you, let me take a step back, because to make it understandable, you need to know something. If you see eye histology, tissue sections of human eyes, taken at different ages, however the specimen was obtained, whether it's a road accident for a 20-year-old who unfortunately had an accident while speeding or whatever, or a pathology of some person who lived to be 85 and relatively good health, you will see dramatic changes in the nature of the outer lining cell of the retina, so-called retinal pigmented epithelium. That cell is a kind of housekeeping cell. So the retinal pigmented epithelium, that is in effect the outermost layer of the retina, is responsible for some essential qualities in cell maintenance and metabolism. And it wearies. It gets tired over time. It can't do its work properly. One of its tasks is the daily ingestion of worn-out parts of the rod cone, the photoreceptor. And it has to ingest these worn-out parts, digest them, and that digestion process is remarkable. And the RPE cell, as it's called, does a wonderful job. But it wearies with time, like all things. And so you have in the human, if you see age-specific tissue sections from human eyes, a gradual accumulation of debris, and actually a slight enlargement of the cell over time. That debris accumulates to a remarkable degree and can disable the cell. There are specific conditions in which there are metabolic defects in which this problem is enhanced and occurs at an earlier age. There are so-called juvenile macular degenerations. But again, if your focus is on age, the cell gets tired. It can't do its job of recycling this spent material as well as you get older. And as I said, the failure to properly digest these ingested materials leads over time, whether it's 70, 80, 90, 100, to true failure, true deterioration of the RPE cell, and in effect to what really now is almost a plague of age-related macular degeneration that you see. There are some early signs of this cell under stress, and if you go to your eye doctor and ask for a checkup and say, you know, I'm 20, I'm 40, I'm 60, I'm 80, can you do a quick snap check? How's my retina? Well, one most likely to say, you know, let me do it right. Let me, you know, do a full examination. But if you say, gee, I'm in a hurry, and, you know, we're old friends, just give me some sense. Can't you do that for me? Well, that eye doctor will say, okay, here, sit down in this chair. This is my examining room. Look at the distance. There's the eye chart projected. Please read. Read down from the top big letter, the big E. So you go and usually cover one eye, test one eye at a time, and you'll go E, Z, B, N, C, D. Read down the lines, and yes, good, okay. You're 20/20 or just off 20/20, but very good, very good. Now, cover one eye. Let's test one eye at a time. Here's my pen light. Look at it. Look at it. Right at it. No, right at it. Don't look away. Look right at this pen light. Okay? Count to 10. 1, 2, 3, so on. Okay. Now, read again. And you'll wink and you'll blink. You're blinded. And 10, 12, 15 seconds, whatever, recovery occurs, and you know, oh, okay, I can see now. E, Z, B, you start reading down, and you recover. Well, if you're 60, 70, 80, and your retina does not have the vigor of youth anymore, you'll find that to the so-called, and this is called the light stress test in the clinic, the time for recovery after looking at a bright light is prolonged. And it may be relatively mild. Instead of 11, 12 seconds, 14, 15 seconds. Well, if in the clinic you don't know anything about a patient, and you just come into a room and you say, Mr. Jones, okay, they've tested your vision already, but I'm Dr. Whomever. I want to do a special test. Cover your left eye. Now, look with your right eye. The eye chart's on. Can you read? Read down to the smallest letters you can read. And the fellow says F, Z, B, whatever line they just say it's 20, 30. You say, okay, now look at my pen light. Look right at it. Look directly at it, please. Okay? Now, read again. And the person looks and often let's get angry. You blinded me. What did you do? I can't see anything. Okay? And so you take an elderly person, especially one that might have some early signs of macular degeneration, and the so-called photo stress test is disturbing because the time for recovery after looking at a bright light is greatly prolonged, and it can go into minutes. And as I said, if one angry patient, you've, as the eye doctor, have derived some essential information, but you haven't made a happy camper out of that person. But you've got your result. And that slow failure that you see with age, especially if there are signs of macular degeneration, is a sure sign that the cooperative function of the essential layers for visual recovery after looking at a bright light, the RPE cell, retinal pigmented epithelial cell, and the photoreceptor, and their cooperative relationship is not what it was in youth. And there's very little to do in the way of therapy, but the point that I think is of real relevance to our present conversation is there are some early signs, and I think they're convincing signs, that the pathologies associated with aging are slowly being unraveled. The mystery of aging is partially understood, and there are real advances in that type of study. Well, again, think to yourself that you are in a pharmaceutical company, you're an authority, and as soon as there's any window of opportunity towards some potential treatment to improve the function of, in this case, the retina, but really of any long-lasting cell of the body that has deteriorated with age, you would get on it with vigor. And so my expectation and belief, with no inside information of any type, is that there are active groups at major pharmaceutical companies working on just this type of cell pathology, trying to see ways to either impede it, reverse it, fix it. And I also am convinced from some studies I've seen that there are likely to be ways to help that, in effect, that there will be some potential therapies, whether they'll be major and revolutionary or minor and simply slow things a little bit, I can't tell you. But I am convinced, and again, with no inside information of any type, that efforts are being put forward at the moment in this direction, and they're likely to have at least moderate success.